Our experiment was on gut microbiome. Our abstract. The gut microbiome consists of microorganisms present inside the gut of an animal, such as bacteria, fungi, and viruses. A microbiome is simply the microorganisms in a particular environment. The gut microbiome can determine factors that lead to health conditions, such as Crohn's disease, obesity, autism, and allergies. A comparison between our two animals was they were both flat-coated retrievers. They both weighed 50 pounds, were female, and had very good health, with no known health issues. They both had the same environment, diet, and exercise. The differences between the two animals was that animal 671 was 1 year old, animal 672 was 5 years old. 671 was unaltered and 672 was altered. And 671 has grass supplements while 672 does not. A brief summary on our experiment. Animal 672 had a large microliter concentration than animal 671. Animal 671 has a larger number of overall reads, which leads to higher overall reads in the kingdom, order, and genus levels. Animal 671 had a greater number of unclassified number of reads at all the same levels. Our conclusion was overall our hypothesis was supported because the gut microbiome of the two dogs was different based on our calculations and experiments. One psychological condition that has been found to have a relationship to the gut microbiome is Crohn's disease. The research that demonstrated this relationship focused on analyzing the gut microbes and amino acids in patients with Crohn's disease. This led to the realization that there were higher levels of amino acids in stool samples from the people with Crohn's disease, which connected with high levels of protobacteria. Researchers then tested on mice to find more details about the relationship between the gut microbiome and these amino acids. With knowledge about how some bacteria in the colon uses urease, an enzyme, to produce pneumonia, they were able to find that urease also allows gut bacteria to produce pneumonia, which is used to make those amino acids. Based on this experiment, the relationship between the gut microbiome and Crohn's disease was causal because it was one specific enzyme that was causing high levels of amino acids and protobacteria. Another psychological relationship is with obesity. This was done with germ-free mice. Given the information that conventionally reared mice have a higher body fat content than germ-free mice, led the researchers to transplant the distal gut microbiota from the normal mice into the gnatobiotic mice. This led to 60% increase in body fat within 2 weeks, even though their food consumption and energy output stayed the same. By Davis Cindy. The gut microbiome obesity continued. The differences between the gut microbiomes was that the transplanted microbiota increased caloric release from dietary plant polysaccharides and modulated host genes that affected energy disposition and adipocytes. This means that the microbiota did indeed have an impact on the obesity, and the relationship is mostly correlative because one thing leads to another. Cindy Davis. Our question was, will a difference in age affect the gut microbiome of two dogs of the same breed with the same weight, sex, health, environment, diet, and exercise? Our hypothesis was, if there is a difference in age between two dogs at the same breed with the same weight, sex, health, environment, diet, and exercise, then there will also be a difference in the gut microbiome because as things age, there's a loss of diversity in the gut microbiome due to several factors, including diet and medication. Our materials and methods. The animals that were compared were two female flat-coated retriever dogs that were both 50 pounds. One was 5 years old and the other was 1 year old. The Cajun kit was used to purify the DNA from the fecal matter. We followed the instructions as the Cajun kit to wash the residual impurities away while allowing the DNA to bind to the membrane. The Zymo kit is then used to concentrate the DNA after being purified. We then follow the instructions of the Zymo kit to concentrate the DNA by adding a elution buffer, incubating, and centrifuging the sample. To find the concentration of the DNA, we use a UV spectrophotometer. We put the DNA sample in the spectrophotometer at a UV range of 260 nm to find the absorbance, to then calculate the concentration in the solution. The amplicon PCR is amplifying the 16S ribosomal RNA gene. This has 3 steps that start with denaturing at 94 degrees Celsius, then annealing at 54 degrees Celsius, and lastly extending at 72 degrees Celsius. There were 30 to 40 cycles of the 3 steps. The gel electrophoresis is used to check for the products of the PCR reactions. We used 250 volts. We carried out the procedure to find any products made by the PCR reaction. The Agincourt AMPUR-XP kit was used for the PCR cleanup. We followed the instructions as stated to remove the amplicon from the PCR reaction. If these were left in the solution, they would interfere with the indexing PCR primers. We removed Mg2+, DNTPS enzyme, and forward and reverse primers. The indexing PCR has 8 cycles of 95 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, and 72 degrees Celsius for 30 seconds. The Agincourt AMPUR-XP kit is used again to remove the PCR buffer, unused forward and reverse primers. The fluorometric assay was used for the quantification of the DNA to determine the concentration of the amplified sample. We use the fluorometer over the spectrophotometer because the spectrophotometer prohibits quantification of DNA and RNA. The fluorometer is more sensitive and specific to nucleic acid interest. The DNA sequence is determined by the Illumina MISC DNA analyzer. This tells the sequence of each index PCR product bound to the chip. Indexing PCR is when you add a set of sequence tags at the end of each amplicon from the sample. This is relative to sequence DNA because we will know which sequence came from which samples from the tags. Our samples show that our sample 671 had 2.35 microliter concentration, and for the 260-280 ratio, we ended up having a 2. For sample 672, we had 0.96 microliter concentration and a 1.91 ratio. For our positive control, we had a 23.2 microliter concentration and a 1.96 ratio. For the negative control, there was no DNA present, and there was a 0.58 ratio. Our gel electrophoresis shows the bands of where the DNA is present, and for our personal results, we only got bands for our positive control and group 586's DNA. Based on the results from the gel electrophoresis, the only samples that had activity was a sample from another group, 586, and the positive control. Since neither of our samples, 671 or 672, received results, our data was inconclusive. The indexing PCR, since the only samples that received results from the electrophoresis were the other groups in the positive control, we did not have enough PCR product to go onto the sequencing step on our own samples. The total reads, this shows our sample 1, 671, we had a total of 20,074 reads and was 100% passing. Sample 2, which was 672, we had 9,200 and 67 total reads and 100% pass. Our positive control had 37 total reads and it was 100% pass, and our negative control was 14 total reads and 100% pass. Our kingdom results showed that 671 had 99.95% bacteria and 0.05 other. Sample 2, 672, had 99.54% bacteria and 0.46% other. The positive control had 100% bacteria and 0% other. And negative control had 100% bacteria and 0% other. This shows that 671 had higher bacteria and higher total reads. Our order results, it varied for each sample, but overall, 671 had higher in each category. Our genius results was also varied for each variable, but overall, 671 was higher in almost all categories. Other and unclassified. For our results in the taxonomic levels, kingdom, order, genus, the sections that were categorized as other were the sum of all classifications with less than 3.5% circumstance, which means that they're considered unimportant. The sections categorized for unclassified are most likely bacteria from a group of organisms believed to have come from a common ancestor base and sequence similarities, meaning the reads had errors. Our discussion. The DNA extraction was successful in terms of quantity, which is important for moving on to the next step. Having a high enough concentration of DNA means that it was pure and did not have contaminations from other proteins or RNA. The positive control final result was 18 microliters, and the negative control was 23 microliters. Animal 672's result was 22 microliters, and 671's result was 20.6 microliters. These results were good and meant that the DNA was pure and at high enough concentration to continue to the next step. After amplicon PCR and gel electrophoresis, the negative control, which we did not add any isolated DNA to, had no product band showing, which was an expected result. This meant that there was no cross-contamination from other sources. In the positive control, we did include DNA, and it had a product band which was expected result. Positive controls were done because if we did not do them, then we would not know whether there was a problem with the extracted DNA, or if there was a defect in the protocol regnans. Animals 671 and 672 did not have a very visible product band. Having no product band for the animals while having a product band for the positive control means that there was a problem with the DNA isolated from the scat samples. Since there was no product bands for animals 671 and 672, we were unable to continue the indexing PCR for them. After indexing the PCR for the negative and positive control, we found that the negative control had no results. This was expected as there was no DNA added to it. If there was a value in the negative control, this would have meant that there was cross-contamination. Since the positive control had DNA in it, we were able to index the PCR, as expected. If we were unable to index the PCR, this would have meant we messed up a step. This also furthered the idea that there was an issue with the animal scat, since we were unable to index those at all. The result for the concentration of the positive control was 63.9 nm, and this meant that it had enough PCR product to go into the sequencing step. When indexing PCR, the minimum amount of PCR product needed is 5 UL microliters. If we do not have this amount, then the concentration will be too low and will not be able to complete the step. We are still discussing animals 672 and 671. Our positive control had 2,816 reads, 100% passing quality filtering, which is an expected result. The types of organisms identified in the kingdom level were 99.96 bacteria and 0.04% unclassified. This is good. It means that there is not a lot of errors or cross-contamination. At the species level, there were 19 total species level taxonomic categories identified. Escherichia alberti made up 79.47%. Unclassified at species level made up 10.26%. Serrata Entophila made up 8.56%. Enterobacter and Asci made up 1.10%. Campylobacter and Candidase and E. coli made up 0.07%. And lastly, Holocroton and Selexogens and bacteroids made up 0.04%. At species levels, it is expected for there to be a higher level of unclassified reads. The results from the species levels tell us exactly what identifiable bacteria was found. It is generally too specific for effective comparisons of animals because of how high the classification portion is. Animals 671 and 672 had 100% read passing quality filtering. This is expected and means that it is adequate to base our analysis on. At the kingdom level, 671 had 99.88% total reads of bacteria and 0.12% unclassified reads. 672 had 99.95% total reads of bacteria and 0.05% unclassified reads. At the order level, 671 had 29 classifications and 672 had 25. Out of their top 8 identified classifications, 6 of them were the same kind of bacteria with different read percentages. The differences were that in their unclassified, 671 had 3.47% reads, 672 had 2.06%. Animal 671 had 2.44% enterobacterials, and 672 had 1.67 campylobacterials, which were the bacteria not shared in their top 8 lists. At the genus level, 671 had 65 identified classifications and 672 had 56. Out of their top 8 identified classifications, 4 of the bacteria identified were the same, but had different read percents. Their differences were that 671 unclassified was 11.75% and 672's was 6.57%. 671 had 7.78% fecal bacterium, 7.25% flavobacterium, and 4.9% cetobacterium. 672 had 5.49% Pseudarella, 3.35% Megamonas, and 3.12% Anaerobacillarium. One type of bacteria found at the genus level of 671 and 672 was Protovella. Protovella has been known to cause issues in the human mouth. It has also been identified as causing abscesses, bacteremia, wound infections, bite infections, genital tract infections, and periodontitis. Periodontitis is a serious gum infection which damages the gums and can cause destruction of the jawbone. Altogether, our animals' data differed very slightly. Their read percents were very similar, and they shared many of the same kinds of identified bacteria. One of their main differences was the numbers of identifiable gut microbiomes, as animal 671 generally had more. 671 was 4 years younger than 672, and so the fact that 671 had more identifiable gut microbiomes helped to support our hypothesis. It also makes sense when taking into account that They share everything in common besides their age. They are mainly similar, but this is the one difference and is important to help to support our hypothesis that age will change the gut microbiome of animals.